PhD from Indian Institute of Science, Bengaluru. He is a postdoctorate from Hebrew University, Jerusalem, Israel, and the Scripps Research Institute, La Jolla, USA. The main focus of his research is to understand the structural principles that govern the sequence to structure relationships in proteins and ligands. He is awarded with National Bioscience Career Development, Rajiv Goyal Youngest Scientist Prize Award in Life Sciences, and many more prestigious awards. Please, sir.
Good afternoon, and thank you for the kind words of introduction, and let me thank Professor Sharma and Professor Panda for giving me the opportunity to come and talk here. But I am aware that we are running late, and I am standing between you people at lunch. What I will try to do is I will give you a quick overview, and maybe those of you who are interested, we can discuss during lunch or afterwards. So what I am going to talk is some of the computational methods which we are developing to address few questions relating to microbiome and things like that. As you heard in the previous talk, for precision medicine, you require genome sequencing or things like that. But now, microbiome is attributed to almost many these things. So now it is not only our genes, it is the genes which make up the entire microbiome, so that goes to the millions. So now, since there are so many different things, microbiome has been implicated. So now we are getting the complete genome sequences from the microbiome, how we can use it to understand the things and design better experiments. So the way microbiome affects many different things, but the language through the language of small molecules are the chemistry, and these are the three ways it can synthesize certain molecule, biosynthesize de novo, it can modify a host-derived metabolite, and it can also do a xenobiotic derived, either a molecule, small molecule which comes from the diet or the drug, it can modify them. And there are experiments coming to address these questions, but with huge volume of data, what sort of computational tool we can develop to use this whole genome of the microbiome and address these questions better. So these are the tools which initially have been developed for the genome-guided natural product discovery. Like natural product discovery was initially a domain of chemists, microbiologists, but with genome sequences came, so there are bioinformatics developed, and same tools can be used to answer this question which I am proposing, and all this involved dealing with sequence, structure, function, on which Dr. Sastry gave a very good overview. And the type of molecules we are looking at is polyketides, non-ribosomal peptides, and a class of molecules which are called bacteriocins or antibiotics, but now their diversity is so much, they are being like, it is a molecule like nisin. It is a peptide which is made in the ribosome and post-translationally modified, and these are being called RIPs, ribosomally synthesized post-translationally modified peptides. So now, and coming to this, then this is the area which is also equally important, like this is the example from digoxin. So a microbial gene which is coming and modifying it so the drug becomes inactive. So as more and more examples coming from experiment, as computational biologists, if you analyze that data properly, we would be in a position to develop the tool which we can use better to predict these things, and that is the purpose of this talk. And this is just a quick overview how, I mean, it looks really challenging that how can you analyze genome, their genomic sequences, and predict chemical structures of the small molecule. But if you take the analogy, if you get enough examples of protein sequences which go to certain 3D structures, you see the commonality or difference between them, you are able to predict structure, and machine learning is helping there. And same thing can happen here. Like what is shown here, these are different natural products. It may be coming from a bacterial genome or microbiome. If you look at their biosynthetic genes, there is a good degree of commonality, and only where there are differences which are shown in the red or the yellow color, those parts of the molecules are changing. So if you keep track, more and more experiments coming and this track, so you can derive pattern and you will be able to predict that. And this is where, like the conventional ideas of bioinformatics, biophysics was helping earlier, and now machine learning is going to help further. And this is where, like this is area we have worked for many different years. There are many programs here, and some of these programs are developed like SBS PKs to look at polypeptides, non-ribosomal peptides had been developed in our group. A challenge was, if you analyze the genome sequence, predict the catalytic domain structure, what you will be able to predict is probably the linear structure of the metabolite, but it can cyclize in many different ways. So now a chemist will be able to look at it and tell this is the correct cyclization, others are wrong. But when you are dealing with big data, and this is where the machine learning is helping, if you give examples what is correct, what is incorrect, machine is able to learn the patterns. And now if you give a genome sequence here like this one, and instead of telling which genes, this is a completely automated prediction of metabolites which are encoded by the biosynthetic gene clusters. I will skip the details, like all the chemical fingerprints and things like that has gone into it. In the interest of time, I will skip all these, and these has to be validated in different trust validation and things like that. And this is we have published in earlier from our group. And there is another class of molecules which I gave you the examples. These are called RIPs. These are ribosomally synthesized post-translationally modified peptides. So what is happening here is, these are small peptides. They do not come from the non-ribosomal pathway. They come from the ribosome. But there are modifying enzymes which are putting cross links, and many different cross links are coming. So to do that, I will skip the pathway. Like what is happening is, like Nissin, this is an example which is a food preservative which is used, and so this is the peptide. This has only many cysteine and serine, and using the enzymes, the serines or threonines are get converted to DHA or a DHB, and another cyclase joins the cross, puts the cross links, and you get this complex chemical structure. So if you look at it, theoretically you can assume that any cysteine will join to any serine or threonine. So you can theoretically say these are the cross-link patterns, but there are, if you go to the examples which are characterized, you will get the correct cross-link pattern. Then it becomes easy to train a machine learning model which can train which are the cross-links which are occurring in nature, because there are certain types of structures are preferred. So that is where you will be able to predict the chemical structure of these cross-linked peptides. And now, if you do that, and this is what we have done in our group, and now these are the open source software which Dr. Sastry was referring to. So now, if you do that, then of course now we are in a position to go to the genome. I will skip this, and to the microbiome, and try to answer questions. What sort of molecules we can predict in the microbiome. So this is what happened when we developed these tools. This is the paper which came in Nature. So this is a molecule like which was effective against VRE, and this was coming from a RIP pathway. It was a Nissin-like molecule. So obviously this was published, but we took our software, tested it on the genome, and we could see that we are almost able to predict the same compound which they had found through experiment. And this encouraged us to look into the whole microbiome, and there are different types of microbiome data. Some are isolates, these are complete genomes, or some will be MAGs, metagenome assembled genomes. So using these, you will be able to see what sort of natural product or the RIP repository it is making. So there are different types of RIPs are listed in this, the lanthipeptides, head-to-tail peptides, lysopeptides, glycosin, botryomycin, cyanobactin, and which types of bacteria are making which types of molecules. So you will be able to predict in an automated way. But the question is, just prediction itself is not enough. If I want somebody to do experiment, I have to tell what sort of function it has, at least even in a prediction way. Then probably somebody can do experiments. And of course, as anybody will do when you have used data, first you do cluster them and see how many different clusters. Now you take all the known RIPs for which we know the function and cluster them. So what you find is the ones which cluster with known RIPs, at least you get a functional clue. And this is the type of analysis we have done. And for many groups, you will be, but the percentage is not, probably about for 20% of them, you will be able to predict some sort of a function, because they are making some sort of molecules for which we already know, and for which we know the function. And this is the reason for this. Then the question here is, like another thing, the ones which we do not cluster, how do you think these molecules might be really made or they are artifact? But if you look at the sequences, these are very small peptides, but if you see the conservation is very high, that is what is shown in this one. Whereas corresponding biosynthetic genes or the proteins are very diverse. So despite divergence of the biosynthetic enzymes, if this short peptides is having such a high degree of conservation, there must be some basis. Probably it is linked to, it has to maintain some sort of a cross-linked structure and some sort of a function. So with that idea, so our tool RipMiner genome, it can run through the whole microbiome and it will be able to predict these types of molecules, and wherever it maps to a known molecule, it will be able to predict the function. And same thing we have done for lasopeptide or other classes where things have been done. So this is a summary of all the like microbiome. These are different bacteria on this axis. These are the different class of RIPs, lanthipeptide, lasopeptide, thiopeptide, and wherever you have a black colored dot, that means we don't know the function, or if there is other color things, that means you can associate some function like pore formation, inhibits food spoilage or cytolysis, whatever function is in literature. So this gives a way to take the whole microbiome genomic data and run these steps of a computational analysis and assign some sort of a function to some of these. So this is next thing I would like to give just few quick slides on. Can we address this sort of a question, like where a microbiome derived enzyme is coming and modifying a drug? So obviously we have to be guided by experiment. Some of the experiments came from literature, like people took different bacterial strains, tested it on different drugs. They found that 95 are not metabolized, there are this many are metabolized. And there are many interesting experiments that took from a microbiome, this particular bacteria, gene fragment, cloned them into E. coli and tried testing which drugs are modified and which drugs are not modified. So with this data, today there is about 25 to 30 enzymes which are known, which are coming from different microbes, and they are introducing modification on different drugs. So that using this data, so now you are in a position to, these we call XME or genobiotic modification enzymes. So they can potentially modify it, and the type of modification, it could be a nitroreduction, hydrolysis, this is known, like even your cytochrome 3 metabolism, how drugs are modified, these are host enzymes are modifying. Only difference here is these are coming from a microbiome derived enzyme, and these are the modification it is doing. So now using this database, so if you scan through the different bacteria, so what you can assign is which class of microbes are hosting these modification enzymes and what they can do to different types of drugs. And this is a static point, and as more and more drugs, like example will this, you can this, but at this point it is a straight cut matching, which is standard bioinformatics tool, this one. But as we have more data, we will be in a position to use machine learning as I used for the example of RIP. So you do not have to go with just the assumption that the same drug is being modified, because the modification is happening on a functional group. So you will be able to predict what sort of modification, which types of enzymes are doing, and even if this a particular drug which is not covered in the experiment, you might be able to still predict if the volume of data is large, and with machine learning, what sort of modification a microbiome derived enzyme will be able to make. So this is the point I wanted to make, and as Dr. Sastry was telling that we all try to prefer have these as open source software. So all these are in the open source. This is not published, but this is microbiome MedDB. So we have a database of this, and somebody gives a genomic sequence, a microbiome derived sequence. It will be analyzed, and either the modification or the enzymes, the de novo biosynthetic whatever it is making will be predicted. So this is the summary of my talk. So what I tried to essentially cover is evolutionary analysis of known natural product biosynthetic pathways in combination with protein structure modeling can help predictive rules for correlating sequence of secondary metabolite gene cluster to chemical structure of their biosynthetic product. Combination of bioinformatics with computational chemistry and machine learning approaches can help in discovery of new pathways, and analysis of rips in microbiome using rip miner genome, this is the tool we have made, helps highly conserved chemical scaffolds, and functions can be predicted for some of them. And using information about genobiotic metabolic enzyme, we have predicted genobiotic metabolic capacity of the human microbiome. So I would stop at this point and take few questions, and obviously those who are interested, we can have discussion over lunch.
Thank you, Dr. Mohanty. It's open for discussion. Any quick questions? Or we can have...
Sure.
That way, the disulfide linkage it will form. It will depend on that.
No, that under the physiological conditions, it will maintain the disulfide bond. If we change it beyond that, the disulfide bond will be reduced. Yeah. No, these compounds are known to be stable. I mean, this sort of things, they don't get reduced by small change in the pH or things like that. And this is how they are doing their function. And these compounds have all originated from when a bacteria is surviving the natural niche. It is the chemical warfare agent of the bacteria. It uses it to kill other bacteria so that it can use the resources better. They are fairly stable compounds. As you know, what you said is right. Some cases, these things can change with environment, but there the design is different, or in a laboratory condition, we change it, go from a non-physiological range, then you can reduce the disulfide bond or make changes to the structure.
Okay, thank you, Dr. Mohanty. So I would request Dr. Shashibala to come on stage to give a token of appreciation to Dr. Mohanty, please.
Please come, please come.
Nowadays it is gaining a lot of importance.
I also request please tell. Thank you very much for all the speakers for their wonderful presentations, and it is a time to honor our chairpersons. I request Dr. Sasibala Singh to honor our chairpersons, Dr. Tikku and Dr. Abhi Sangambor. Okay, thank you very much for all. So we break for lunch. So we will reassemble by 1:30 sharp for starting the session, next session. So yes, we have electric functions afterwards, and we will be starting in time. Thank you.